So now that we've discussed the oxyhemoglobin saturation curves for variations in adult physiology, so changes in pH, changes in temperature, etc., we also have to talk about fetal hemoglobin. So hopefully by now you've had a chance to at least look at the differences in fetal circulation from the last chapter. So bypassing of the lungs, interfacing with the placenta via the umbilical arteries and vein. The one thing that is not explained by fetal circulation is how the oxygen is transferred from mom to infant. And that's a challenge because, if you have hemoglobin with the same binding affinity and you have the same temperature and pH, what's to move the oxygen from mom into baby's blood? One thing is the partial pressure gradient. So fetal venous blood is lower in partial pressure of O2 than maternal arterial blood, so there's that. But also, fetal hemoglobin is a little bit better at hanging on to oxygen, so it tends to grab it and bind it more tightly. So that is beneficial to the fetus because it means that for the fetus, its blood is quote unquote stickier to oxygen than maternal blood. Placental blood has a partial pressure of oxygen of about 30 millimeters of mercury. So if we go to 30, fetal hemoglobin's affinity or percent hemoglobin saturation at 30 is much higher than adult hemoglobin. So we have hemoglobin releasing oxygen here, and it moves to the fetal hemoglobin, and that's how the hemoglobin is transferred, which is fascinating. That's why I think fetuses are so cool, because we think of them like, yes, they're humans that are still forming. But in a lot of senses, they're creatures that are very unique because they have a lot of properties and talents and physiological differences that adults don't have. So they're fascinating from that perspective, as well as being cute. So carbon dioxide transport occupies a lot of this PowerPoint and a lot of your textbook because oxygen binding and unbinding from hemoglobin is pretty straightforward. We just looked at the curves for the things that influence the rate at which hemoglobin is going to bind or let go of oxygen. But a lot of that stuff is driven by the chemistry of carbon dioxide. And carbon dioxide is also an important former of the buffer system in the plasma. So remember, buffers are solutions of a weak acid, in this case, carbonic acid, and its conjugate base, which is bicarbonate. So carbonic acid is in equilibrium with its form as it splits, and this arrow has two heads because depending on the relative amount of reactants and products, or vice versa, the reaction can shift to the right or the left, and that avoids large changes in pH in the body. So although we consider carbon dioxide to be a quote-unquote waste product of metabolism, it actually has a really important role in regulating our body pH. So we have to talk about carbon dioxide transport for that reason. So there's three modes of it. Most carbon dioxide is not transported on a molecule, so it's different from oxygen in that way, but rather in the form of bicarbonate ions. So you convert your CO2 into HCO3-, and it exists hidden in this base. Carbaminohemoglobin is the other 23%. So this is a long, complicated word. It's not hard to understand, though. So keep in mind that hemoglobin is a tetramer. It has 4 amino acid chains all folded up, and then heme groups. The oxygen only binds to the heme groups, which means that there's other open real estate to bind other things. Carbaminohemoglobin is called that because the carbon dioxide that's transported on hemoglobin is not bound to the heme groups, it's bound to the amino acid chains. So the amino part of that word reflects that property. And then about 7% of it is dissolved in plasma. So compare that with oxygen, and you can see that carbon dioxide is a lot more soluble in plasma than oxygen. So oxygen is like 1-ish%, and carbon dioxide is 7. So CO2 is diffusing out into the bloodstream. 93% of it stays in the red blood cells, only 7% remains in the plasma. Of the 93% that enters red blood cells, 70% undergoes the carbonic anhydrase reaction and turns into protons and bicarbonate. The other 23% sticks to the non-heme part of hemoglobin. Now, this part is interesting, but I'm going to discuss it on the next slide. And in order to do this, I'm also going to employ the use of my handy-dandy magnifying glass because I want to focus on one side at a time. Let's do this one. Okay, so we are looking at an actively respiring tissue, and that is unloading hemoglobin. So keep in mind, here we have unloading carbon dioxide. We've got carbon dioxide diffusing from the tissue cells into the bloodstream. The little arrow going down into the plasma outside of the red blood cell, this is going to be that 7% that ends up dissolved in the plasma. The rest of it, 23%, is going to just hop onto the amino acid chains of hemoglobin. The rest of it is going to undergo the bicarbonate reaction, the carbonic anhydrase reaction. So we have H2CO3, carbonic acid, converted into bicarbonate and a proton. Both of these things are important. So let's start with the proton. The proton is going to increase the acidity. So the more of this leg of the reaction that's happening, the lower the pH in the red blood cell is going to be. Think back to the oxyhemoglobin saturation curve. The lower the pH, the more acidic, the lower the binding affinity for oxygen of hemoglobin. So this proton actually functions to lower hemoglobin's affinity for oxygen, which if you look up here, and I wish I could drag my little magnifying glass around, but apparently I can't. This guy. Well, I guess I can if I just hold. So if I go up here, I can see that hemoglobin is letting go of oxygen, and oxygen is diffusing by itself, not attached to hemoglobin, into the tissue because it's higher in partial pressure in here than it is in here. But this part of the drawing does not address what actually makes that happen. So the question is, how does the hemoglobin and the red blood cell, quote unquote, know when the right time to drop off oxygen is? And the answer is, because of metabolic tissues producing carbon dioxide, which turns into protons and bicarbonate, when a red blood cell is in the neighborhood of a tissue that is actively respiring and performing metabolism, that red blood cell is going to get increasingly acidic. And when that red blood cell gets increasingly acidic, more oxygen will hop off the hemoglobin, and interestingly, protons take its place. So the proton hops on and basically boots the oxygen off. The other component of this is the chloride shift. Now, this is important. It also has a funny name, not chloride shift. Chloride shift is a name for it. It's not the funny one. The funny name for it is the hamburger shift, because a guy named Dr. Hamburger discovered it. So if you see the word hamburger shift, it means this. So here's the deal with the blood buffer system and resisting pH changes in interstitial fluid. So now you know from capillary exchange that interstitial fluid comes from the blood, yes? Bicarbonate is an important buffer for both plasma and the interstitial fluid. It's only useful in that role if it's not stuck inside of a red blood cell. So we have to find a way to get the bicarbonate out to where it can be useful. Here's the rub. Cells, all of your body cells, not just neurons, not just muscle cells, all of your body cells work hard to be more electronegative on the inside than the outside. And this is for a variety of reasons, most of them related to membrane permeability and transport. So if we want the bicarbonate to be out here in the plasma, if it were to just be leaving in droves, this cell would be hemorrhaging negative charge. It would be losing all of its cations, and that would change the gradient across the membrane and change the permeability of the cell. We don't want that. So if you are going to be putting bicarbonate out, you have to exchange it for another minus sign. So the chloride shift is just a simple exchange from one anion, bicarbonate, for another, chloride. So it's a way for bicarbonate to get into where it's useful without changing the net charge across this cell's membrane. Nifty, huh? Okay, let's go over to this side, where everything basically happens in reverse. So we've got reverse chloride shift, and this is driven by the following. The alveolar airspace has a lower partial pressure of carbon dioxide than inside of the red blood cell. So 45 here, 40 here. And the partial pressure of oxygen is also different up here. We'll go up here in a minute. So we have the reverse chloride shift taking place, where due to the changes in chemistry inside of the red blood cell, now we're pumping chloride out and taking bicarbonate in. Additionally, because the binding affinity for oxygen is changing based on the changes in chemistry and partial pressures near the alveolar airspace, hemoglobin parts ways from that proton, and that proton is now free to recombine with bicarbonate, forming carbonic acid. If you take a water off of that, using again carbonic anhydrase, you get CO2. Now we have soluble CO2 building up at the red blood cell and able to diffuse freely into the alveolar air. So if I go up here, we can see that the partial pressure of alveolar air, which remember is 100 millimeters of mercury, is a lot higher than in here, where it's about 40. So there's a 60 millimeter of mercury difference. That's a lot. So that's going to drive oxygen just by diffusion into the cell. And because of the cooperative binding effect, once that first oxygen binds to a hemoglobin, it's more likely to pick up a second. And once it's picked up a second, it's more likely to pick up a third. So that steep concentration gradient combined with the binding affinity changes favors binding of oxygen to hemoglobin. So this hemoglobin basically is kicking off a proton and saying, get out of here, I want to go bind to oxygen, and oxygen hops on in its place. So if you examine this chart very carefully, what you'll notice is it's two sides of the same coin. So one thing is happening in reverse of the other. And of course, now I can't unmagnify. There we go. So now we'll move on to control of respiration. There's a question in the chat before we move on, and that is the chloride and bicarbonate exchange, is it active or passive, and do you need to know? And the answer is you do not need to know. It is technically active transport, but that antiport is not important here. All right, control of respiration. Why would you need to control gas exchange at all? Well, your metabolic needs differ depending on your physiological state. So, for example, I'm just sitting here talking to you. It's not eating up a lot of my metabolic budget, right? I'm moving my mouth, I'm putting my feet on the ground, it's pretty easy. If I get up and go running, that's going to change really quickly. So my skeletal muscles are going to start demanding lots more oxygen, and I need my ventilation and gas exchange to keep up. Fortunately, I don't have to be in charge of that mentally and voluntarily. I have regulatory centers which control that for me. So local regulation of respiration includes pulmonary capillary diameter and airway diameter. So if you think back to the PowerPoint about blood vessels, we talked about local autoregulation and the fact that changes in oxygen consumption change the diameter of arterioles. So the faster the metabolism in the capillary bed, the wider the arteriolar diameter gets. Same is true of airway. So if I'm going to increase my respiration rate, let's say I'm exercising. Exercise activates the sympathetic nervous system, and sympathetic innervation of the airway causes bronchodilation, which makes my airway wider and easier to deliver air to my lungs. And then with regard to the central control, CNS respiratory centers basically dictate the depth and frequency of ventilation. So how fast you're breathing, so number of breaths per unit time, but also how deeply you're breathing. So are you really drawing in a lot of air, or are you breathing relatively shallowly? Respiration is closely matched by changes in cardiovascular function. And when you look at the diagram in the next slide, that is going to show you the centers that control respiration, you'll get a better view of why that is. So remember the cardioacceleratory and cardioinhibitory centers of the medulla oblongata. You'll find that pons and medullary centers regulating breath are right nearby. So let's have a look at that. So we have voluntary and involuntary control, as I mentioned previously. And the involuntary control comes from the pons and the medulla oblongata. We'll get to the details of that in the next slide. Voluntary control initiates at the cerebral cortex. So remember, the precentral gyrus contains upper motor neurons that are mapped to your musculature. So there's a motor homunculus that is corresponding to the locations of muscles and also how many motor units they have. That's a reminder for 241, hint, hint. So those upper motor neurons, those pyramidal cells, descend and basically override the medullary and pons sections in the short term to change your breath. So let's first have a closer look at the medullary respiratory centers. So the respiratory centers in the medulla oblongata are going to regulate frequency. So basically you have these little populations in the medulla, and they're the dorsal and ventral respiratory group, respectively. So two nuclei meaning cell bodies, right? So the dorsal respiratory group is a group of neurons that is active every single cycle. And if you look at what it innervates, you'll find that its innervation pattern innervates the diaphragm and external intercostals. So here we, for example, can see the phrenic nerve innervating the diaphragm. So if you pay attention to the muscles of inspiration and expiration, you'll be able to figure out that if the dorsal respiratory group innervates the diaphragm and intercostals, that must mean that the dorsal respiratory group is responsible for inhaling. And that's true. So this is active every cycle. So what you would expect to see, as far as if I put an electrode in here, and then I watched what those neurons were doing with each inhalation of a patient, what I would expect to see is a spike train, so lots of action potentials in a row, during every inspiration, and then nothing during expiration. The ventral respiratory group is only active during forced breathing. So if I need to voluntarily breathe out, like I just did, my ventral respiratory group is active. So this is only active during forced respiration. It's going to coordinate the muscles of forced expiration, and it innervates the accessory muscles. So my internal intercostals, my abdominal muscles. There are pacemaker cells in these, specifically in the dorsal respiratory group, that control unconscious breathing. So just like in your heart and in your enteric nervous system, which we'll get to in a little bit, there are cells that spontaneously depolarize. So remember the pacemaker potential from the cardiovascular system, where it was leaky, those cells were, and that caused membrane potential to drift towards threshold all the time. Same deal here. So this is clinically relevant to you, especially if you're going to be a nurse and potentially, administering drugs prescribed by a doctor and monitoring the effect of them on their patients. CNS stimulants and depressants are known to affect pacemaker cells, especially if they can cross the blood-brain barrier. So right now in the United States, we are in the middle of an opioid crisis, and overdoses on opioids often cause death by asphyxiation, wherein someone uses the drug, gets high, and basically just loses consciousness and stops breathing. Why do they stop breathing? Well, ordinarily, if you're asleep, your dorsal respiratory group is still active. If you subject your nervous system to large amounts of a nervous system depressant, that changes the leakiness, that changes the leakiness of the DRG neurons, and the pacemaker effect no longer exists, which means that the impulse to breathe in unconsciously no longer exists. And that is a really, really sad way that a lot of people die in the United States. So, of course, sometimes it's medically necessary to prescribe depressants or stimulants, for example, induced comas. But in those cases, that requires close medical supervision by a medical professional so that the patient is safe. Oh, just really quick before we move on, I mentioned Ondine's curse earlier in the lecture. I said that poliovirus affects neurons, and it's true, poliovirus attacks nervous tissue specifically. So if the poliovirus manages to harm either these neurons or the dorsal respiratory group more commonly, that ends up ablating the involuntary unconscious inhalation function, and that's why those individuals have to breathe consciously. So the Apneutic Center and the Pneumotoxic Center. These are in the pons, so up here. And these modify stuff that already happens. So what I mean by that is, if you're already inhaling and you need to continue, the Apneutic Center is going to continue the inhalation. And if you're already exhaling and you need to continue, the Pneumotoxic Center promotes exhalation. So if you inhale for longer and you exhale for longer, for example, that means that your breath rate is going to be deeper and slower. So these play a role in slow, deep breath. And this is all in response to stimuli. So an example is stretch receptors in bronchioles. So how wide is your airway? How deeply do you need to breathe accordingly? Also chemoreceptors in your carotid and aortic bodies. So both receptors for oxygenation and also receptors for pH. And baroreceptors in the carotid and aortic sinuses. So the changes in breath that you see that are not necessarily all that voluntarily when you exercise. So think about a time when you were exercising and you got out of breath and then you stopped and you were just kind of standing there going, and even if you wanted to slow down or change the depth of your breathing, you couldn't. That's because you're paying back an oxygen debt incurred by your skeletal muscles. And in order to do that, your pons is basically taking over respiration and saying, like, we got to gather more oxygen from the environment, so I'm going to make you do this for a while until the problem goes away. So that's an example of unconscious homeostasis by your autonomic nervous system taking over for you. Interestingly, there's another phenomenon based on this that is medically relevant, and that is the phenomenon of something called Kussmaul respiration. So this is a consequence of untreated diabetes mellitus. And what happens with diabetes is if it's untreated due to chronic high blood sugar and the blood sugar not moving into the tissues, the body begins to burn fat and protein to survive. That releases ketones and causes a condition called diabetic ketoacidosis. The acid there is detected by chemoreceptors, and that information is furnished to the pons and integrated in the medulla. So this happens most frequently in children. You don't tend to see Kussmaul respiration in adults very often. But if you have a kid that's like super duper thirsty, peeing a lot, and is breathing very deeply and very quickly, basically doing a pant, and it's involuntarily happening, that's Kussmaul respiration, and that's a classic hallmark of diabetes. Someone's asking how to spell Kussmaul, and it's a very good question. It's Kussmaul, Kussmaul respiration. All right, so we have run out of time, and we're almost done with the respiratory PowerPoint, but not quite, so I'll have to finish it later. So for now, I'm going to bid you guys a good evening, and I'm going to get to work processing this video so I can get them posted as quickly as possible. So thank you for your attention, and have a great evening.